Right. So this is part 4 of the chapter 17 lecture. Even though this part is not actually in the book, I'll be sharing some ideas and some techniques that the book has not covered, but I think they might be very useful for anyone going into biology. So hang in there. We'll talk about also why is it that we've been talking so much about these technologies, and how is the fact that um, we've been able to accomplish so much without actually doing a lot of these experiments in humans. Next semester, you all are going to be working in Bio 4B. So in that class, you are going to be focusing on the diversity of life from bacteria all the way to humans. And you'll be noticing that a lot of the same genes that affect bacteria affect humans. And that is why it's so important to understand that what happens in a bacteria happens in an elephant, molecularly speaking. So the idea of evolutionary um, conservatism or evolutionary conservatism, yeah, conservative among all the species. Our DNA is very conserved among many of our species. I mean, we're only what 1.5% different than gorillas. Like, what is a 1.5% of our genome that makes us so different than gorillas, that makes us so, I mean, that makes us walk upright, talk, reason, make art? I mean, that is because of DNA and how similar our genome are, and how complex that 1.5% can be. So enjoy this lecture. It'll be a little bit messy, but I hope you enjoy it. See you at the end. Started. Um, what I want to first talk about is um, the difference between DNA work and RNA work. So when we talk about DNA work, we talk about genes, we talk about chromatin, we talk about what else are we talking about? Chromatin, genes, um, and of course genome. When we talk about RNA work or RNA experiments, RNA techniques, we talk about mainly gene expression. Now, I know that you guys want to tell me, yeah, but there's other kind of RNAs. You have ribosomal RNAs, you have um, tRNAs, which are for transcription. You have small RNAs, microRNAs, and all of those. That's fine. But I want to make sure that you understand that these two molecules, these two types of techniques, which are many actually, are very different. Okay, now we talk mostly when we talk about electrophoresis and PCR about DNA, but we didn't really talk about so much about RNA work. So remember, the problem with RNA, or the thing about RNA, is that every single cell, so let's draw here a neuron. So a neuron, um, then you have, what other cells do you have that I can talk about? Then you have your cells in your intestine, right? Every single cell in your intestine, your Krebs cells, your goblet cells, all of those cells. And then you have um, your skin cells, right? So you have layers of epithelial cells in your skin. Now, we talked about during our gene expression about the fact that every single cell has the same DNA, right? However, the genes expressed in each of these cells are going to be different. The genes expressed in a neuron, in a intestinal cell, or in a skin cell are going to be different. So when scientists try to look at gene expression, they have to first isolate the cells or the tissue they want to work with. So, in other words, if we are trying to see gene expression profile of a neuron, we're not going to take a piece of the skin of the scalp, right? We're going to go into the brain. I'm actually not going to try to draw a brain. That's going to be a little crazy. But anyway, and then get a sample of that tissue. So that's a sample of your brain tissue. And then they need to extract the RNA, and then they need to make the RNA into cDNA. And then once that cDNA is done, then they can use it for multiple things. So they can do RT-PCR. If they want to do real-time PCR, they can do qPCR. If they want to do quantitative PCR, just to look at gene expression, they can see in a table, they can check, well, gene A and gene B, they're widely differently expressed, or they can, if they look at brain tissue from, let's say, let's say this is the frontal lobe, and then they get another brain tissue from the occipital lobe. They can see, well, gene A in the frontal lobe is expressed this much, and gene A in the occipital lobe is expressed this much. But gene B in the frontal lobe is expressed this much, but in the occipital lobe is expressed this much. So I'm trying to tell you here that you can actually quantitatively select the genes that you want to study and the tissues that you want to study and check how much they're expressed in different tissues. Okay? Now, you can also do something a little more crazy and a little more expensive, which is called RNA sequencing, where you literally send all your sample to somebody, and after a while, they're gonna give you a file. They're actually gonna give you a file with a bunch of ones and zeros and ones and zeros and zeros and ones, which will give you, you have to run a algorithm to give you a gene profile, or an expression profile. And then you can use multiple languages like R, Batch, Python, they say R already, um, to show pretty much this, right? Now, just imagine you are in a lab where you're trying to find out if you have a drug A, and you're trying to find out if drug A is good for the brain, and then you have a placebo, and you're gonna ask the same question for a placebo. So then you can take the RNA from each of these two groups, then send it out for RNA-seq, RNA-seq. And then you can literally ask, what about gene A in these two groups? What about gene B? Are they different? Are they the same? How many of these genes are similar in the drug A versus a placebo? And then you can start asking, okay, so I find that 100 of these genes are upregulated. So they go, so 100 genes are up, what about those? Like, do they have any similarities? And then you start seeing that maybe some of these genes that are upregulated in drug A are, are important for development. So now that's crazy point number one. There's another crazy point. I know. Oh my God, Dr. AB, shut up. But you will have one more here called single cell RNA-seq. That literally means that you can take. Now I'm gonna try to. I actually don't know how to. Let me do a simple one. I know how to draw the brain of a mouse. So let's say you are trying to study this drug A in the mouse brain, right? Of course, that's been the intention all along. You can literally send the whole brain to be tested. You might have to do some work to get single cells, and every single cell can be coded with a specific mark, with a specific it's called barcode. And then you can literally ask, no use whether or not the whole brain is affected by drug A, but you can ask which of these cells is affected by doing single cell RNA-seq. So this is where we're getting into the metadata as well. Okay? Now, in the next slide, I'll talk about the some of the DNA techniques that we have not talked about yet. So in the book, we talked about this technique for protein analysis, where we had a bait and a prey, and we're trying to see if protein A and protein B interact, and that's fine. You can also do this with something called immunoprecipitation, which I'm not going to go into right now. Although the idea is to use antibodies. Remember, with antibodies, you can actually buy or create antibodies directly designed for your protein of interest. And then you can pull them down like precipitates, like when you put, when you're trying to, I actually don't have a good analogy. I'll get back to you on that one. But you can use these antibodies to pull down certain proteins and ask what other proteins are interacting with my protein of interest. But then you can also have protein DNA interaction. That's why I put it here in this section of DNA work, because there are going to be some proteins, like let's say protein A, that binds to DNA, and you can use what's called ChIP-seq. What is ChIP-seq? It means chromatin. You guys now know what chromatin means, right? DNA, immuno, precipitation. And I just noticed that I misspelled this immuno here. Immuno. Precipitation sequencing. Pretty much you're going to use antibodies for this protein C, and then you're gonna ask what DNA sequence is protein C bound to? And believe me, the DNA is huge, right? You have 23 chromosomes in humans times 2 because you get one from mom and one from dad. And protein C will bind to these areas. So, the reason why we've been talking so much in the previous slides about DNA sequencing and all of that is because everything comes together, everything interacts with each other, everything is interwound. So while we're talking about DNA sequencing in a very overall aspect, we need to remember that it's because of specific techniques that people use. Um, yeah. So we've been studying biology this past semester, learning about all of these molecular processes and discoveries from many, many scientists. But what is the point? I mean, many of us or many of you guys will not necessarily go into hardcore um, E. coli research or hardcore, Drosophila research. Maybe you do, who knows? After meeting with me, who knows? However, many of you are going into the medical field, whether it's for nursing or for nurse practitioners or physician assistants or physical therapists, occupational therapists, and so forth. Some of you may even go into the dark arts, I mean, medicine, right? Haha. But what is the point of studying all of these basic things? So first, I want to introduce this concept or this phrase that it always gets to me, and I paraphrase it many times, but officially it is: what is true for E. coli is true for the elephant. Elephant. This was quoted by or coined by a scientist, a French scientist in the mid 1900s called Jacques Monod. He was actually a microbiologist in France, in Paris, during, funny enough, well, not funny, but weirdly enough, during the time of Nazi occupation in France. He actually was part of the resistance. While they were going on the Nazi stuff in Paris, he was actually doing a lot of experiments on the side. But anyway, so what is true in E. coli is true in an elephant. I paraphrase it by saying what happens in E. coli happens in elephant. It's because in a molecular level, and if you're even more scientist geek, in a physical level, in a chemical level, everything is ruled by the same properties, right? I mean, our water molecules are no different from the water molecules found in E. coli. It is the same water molecule. The same DNA that instructs what to do in E. coli instructs us what to do because everything is conserved. So this idea of evolution and conservation in evolution means that a lot of the same processes, a lot of the same mechanisms, and of course, a lot of the same genes can be found up and down the evolutionary tree. This is why I was able to use Drosophila to study human leukemia, if you remember from my intro. So this also brings me to the idea of model organisms. These are and organisms like animals that have been established in research as homologs to humans, organs, or mechanisms. Mechanisms. So that means that scientists have been able to study these animals, these models, and find a parallel to human mechanisms. For example, Drosophila. It has been studied for disease, for development, for stem cell research. I mean, name it, and they have been be able to use it. C. elegans, little round worms. They have been used for developmental studies, for neurological studies, for stem cell research, for drug development. Of course, you know about yeast. Same thing. My friend from NYU in 2025. So if you're listening to this in 2025, it's just now. If you're listening to this later, it's been not so long, hopefully. He actually studied chromatin modification in yeast. Um, One scientist from the HHMI, which is a huge honor to be a scientist in HHMI, she actually established work using yeast for neurodegenerative diseases. Neurodegenerative diseases, which open up a whole field of being able to study how proteins get misfolded and create diseases like Parkinson. So then, of course, you have our old friend, the mouse and the rats. They have been well studied in different cases. You have pigs. You have then the primate research, such as monkeys or apes, I should say. Then you have, and then of course you have human cells. There are labs out there that use all the research only using human cells. Um, this brings me to the HeLa cells. HeLa cells were created by these scientists taking tumor cells from a cervical cancer patient called Henrietta Lacks, who happened to be a African American woman living in Baltimore, and she never knew that he was doing that, and he became super famous. For the cells that he stole away, pretty much, because back then I think it was in the 60s, nobody was really taking care of sign permissions and much less taking care of minority people. So anyway, that's a whole different point. Um, you have embryonic stem cells. Embryonic stem cells. So literally, cells from from human embryo have been um, harvested and developed into lines. And of course, before the ban in 2001 of a stem cell ban, um, they used to go collect embryos that were going to be discarded and start doing research in them. And then the ban started, and only those cells that have been established. So established means that people have been already using. Um, they were able to use now. I mean, when I was in Stanford, I used embryonic stem cells called H9, which were collected right before the ban. Anyway, so, and of course, we have um, tumor cells and so forth. So many human cells have been done for, having used for model organisms. The point here is that whatever people find in these model organisms can be extrapolated to human. Um, and That is a concept that I hope everybody understands here. And the reason of evolutionary conservatism, or conserve pathways, is very, very true. And I hope in the next couple of slides, as we finish this lecture, you will understand why this is so important, and why, even though we have been learning these concepts in a very abstract way, it's not really abstract, because all of these that you have learned can be directly translated and directly put into use in labs and medical research, clinical research, human research, because everything is connected. In this slide, I want to reiterate how conserved our genome is in multiple species. Hox genes are a group of genes that are used for anterior to posterior axial differentiation. That means that some of these genes are going to differentiate the anterior part of the embryo, while other genes are going to be used for the posterior part of the embryo. However, if you notice in this diagram, you can find the exact same genes involved in similar areas of the fly body. For example, Hox A1, Hox B1, and Hox D1 are all for the most anterior part of the column. They're all used there. And a very similar gene found in flies is also used for this anterior development. And you see this along the side of the anterior posterior region. Now, let's look at embryology to see how similar we are with so many different species of animals. Here I give you 6 individual early embryos of 6 different animals. I'll give you a hint. Only 2 of these embryos are from mammal animals. Could you identify which one of these will give rise to a human being? Boom! What about now? Do you guess correctly? We share similar morphological structures all the way from early embryo, with fish all the way to sheep. Isn't evolution amazing? The conservation of DNA and the conservation of structures. You will see more of these next semester in Bio 4B. All right, so that's the end of part 4 of chapter 17 about technology. Even though this is not completely covered in your textbook, I hope you understood the fact that we humans can only survive, or have survived and have been able to find new ways of medicine and treatment, because we're able to do basic research. And we can do basic research because we can understand, or we do understand, the basic processes of life: cellular respiration, photosynthesis, osmosis, diffusion, mitosis, meiosis, binary fission for those microbiologists among us. All of those processes, cell signaling, how cells talk to each other, is the same. They do the same mechanisms. They are ruled by the same properties, by the same laws. if we understand the basics of biology, we can open our minds to a whole new level of possibilities to learn new things. So I hope you understood that. Next semester in 4B, you're going to have much more fun with diversity and with biodiversity, I should say. I mean, diversity is also good. So please make sure you ask any questions if you can. And I will see you next time. Bye.